Hello and welcome to my YouTube channel, Perfect Lab. So have you ever wondered how microbiologists identify and differentiate bacteria under a microscope? Gram staining is a fundamental laboratory technique that separates bacteria into gram-positive and gram-negative groups based on differences in their cell wall structure. So in this video, I will discuss the principles of gram staining, the importance of proper smear preparation, and the step-by-step staining procedure used to achieve accurate and reliable results in the microbiology laboratory. So I will just encourage you to relax and let's get started. So we'll start our introduction by looking at what gram staining is. So gram staining is a differential staining technique which is used to classify bacteria into gram-positive and gram-negative based on cell wall composition, and this technique was developed by Hans Christian Grams in 1884. So now let us look at the principle, and before we look at the principle, I would like you to know that one of the major differences between a gram-positive and a gram-negative bacteria has to do with their cell wall. The gram-positive has a thick peptidoglycan cell wall, whereas the gram-negative has a thin peptidoglycan cell wall. So the principle is that gram-positive bacteria retain the crystal violet stain because of their thick peptidoglycan cell wall and therefore appear purple under the microscope, whereas gram-negative bacteria lose the crystal violet stain during the decolorization step due to the thin peptidoglycan cell wall, and as a result pick up the counter stain, which is the safranin, and appear pink or red under the microscope. So this is a picture showing how a gram-positive bacteria picking the primary stain, which is the crystal violet, appears as a purple color under the microscope. And this is also a picture showing that of a gram-negative bacteria picking the counter stain, which is the safranin, after losing the primary stain during the decolorization step. So let us look at the gram staining method. So before gram staining is done, the first thing that we have to do is to prepare our smear and feast it. So to prepare a smear, a thin bacterial smear is prepared on a clean glass slide as shown over here, and then allowed to dry completely, after which it is heat feast by passing it through flame 2 to 3 times. And the heat feasting is to kill the bacteria and also to make the cells adhere firmly to the slide, and also to preserve the bacterial structure in integrity for gram staining process. So let's look at the importance of the smear preparation, the air drying completely, and the heat feasting before performing our gram staining. One is that it ensures the bacteria adheres to the glass slides and remain in place during the washing and the decolorization process. So the second importance is that it spreads the bacterial cells into a single thin layer as shown over here to enable accurate microscopic identification. So now let's look at the importance of the air drying of the bacteria. One is that it prevents cell lysis. This is because whenever we heat a wet smear, the residual water boils instantly, and as a result a vapor pressure is built, and this vapor pressure causes the cells to rupture, distort, or destroy them. So 2 is to preserve the bacterial morphology. This is because allowing the smear to dry completely ensures that the bacteria maintain their original size, shape, and arrangement, which is essential for accurate identification. And 3 is to enable proper fixation. And one key thing that we have to notice is that heat fixation works best on a dry smear. This is because a dry smear firmly attaches the bacteria to the glass, preventing them from washing away during the staining and the reagent steps. So now let's look at the importance of the heat fixing. One is to kill the bacteria, and this is to make the bacteria safe to handle during the gram staining process. And two is also to adhere the bacterial cells to the glass slides so that they do not wash off during the staining process. Three is also to preserve the bacterial cells morphology so that they retain their natural shape under the microscope. Now let us look at the gram staining steps, or the various steps involved in the gram staining technique. So the first step has to do with the application of the crystal violet. The second step has to do with the application of the gram iodine, which is the mordant. The third step has to do with the decolorization step, and the fourth step has to do with the addition of the counter stain, which is the saffronate. So now let's look at the first step, which has to do with the application of the crystal violet. So to do this, we flood our slides with the crystal violet, which is acting as our primary stain. We then allow it to stand for 1 minute, then we rinse with water as shown in the picture here. So for the second step, we flood the slide with our LUGO or gram iodine, which is acting as a mordant. And what happens is that the mordant forms a crystal violet iodine complex. And also over here, we allow the slide to stand for 1 minute and just rinse. So the third step, which is the decolorization steps, involves flooding the slide with a decolorizer, which is either a mixture of acetone and alcohol or just alcohol, for 5 to 15 seconds, and then rinsing with water. And the final step, which has to do with the application of the saffronin. In this case, we flood the smear with saffronin, and basically this is to stain the gram-negative bacteria pink and just allow it to stand for 30 to 60 seconds and then rinsing it with water. Now let us look at the various results that occur in each of the 4 stages. So for the crystal violet application, what happens is that both the gram-positive and the gram-negative bacteria picks the crystal violet, which is the primary stain, so they both appear purple as shown here. Now for the addition of the gram iodine or the LUGO iodine, which is acting as a mordant, also what happens is that both the gram-positive and the gram-negative cells still maintains the purple color of the crystal violet. The third step, which involves the decolorization step, happens to be a key step in the gram staining techniques that helps to differentiate the gram-negative bacteria from the gram-positive bacteria. And in this case, what happens is that the gram-negative bacteria, because they have a thin peptidoglycan cell wall, will lose the primary stain, which is the crystal violet, during the decolorization step. And as this happens, they are able to pick our saffronin, which is the counter stain, and turns pink or becomes pink under the microscope. So thank you for watching my video, and if you found this video helpful, please like, share and subscribe to Perfect Lab. Also leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video, and have a perfect life.